your body cycling through nutrients?
Yes, using. That's right. But what does cycling involve? You're on the right track.
Yeah,
Say
that's
more
all I
about
got.
It's kind of like breaking down stuff and then using the broken down stuff to build up other stuff.
Right. So it includes the breaking down of stuff and the building of stuff. What are the names for the breaking down of stuff and building of stuff? So metabolism is all the chemical reactions that happen in a cell. It's divided into two categories: the breaking down and the assembly.
Catabolism.
Breaking down.
Anabolism and catabolism. So catabolism is the breaking down, and how do you break it down? To get energy, right? So catabolism is to break stuff down and get energy. Anabolism is to build things back up again. And the way that I used to remember it when I was trying to keep those straight in my head is anabolism starts with an A, and so does assembly. So anabolism uses the energy that came from catabolism to build stuff back up again. So we're going to talk, today we're going to start out talking about catabolism and how organisms generate energy. Before we do that, we have to do a little bit of thinking about types of organisms, though, and where they get the energy that they're using. This is, what the heck? Where'd that go? That's the wrong one. You guys like the notes on that side, right? This is a figure from your book on page 232 that tells about different categories of organisms. And so far, when you guys have learned about metabolism, you probably were focused on human cells, which don't have nearly the versatility that microorganisms have. And you probably focused on glucose and glycolysis and the Krebs cycle and respiration. We're going to talk about those really briefly, mostly to refresh your memory and give you a context to put some new ideas into. But this, let's see here, I'm going to keep myself honest and actually use the notes here, not just make this stuff up as I go. In terms of energy, microbes can be broken down into a few different categories, and in terms of their carbon sources, and that's shown on this figure at the beginning of the chapter. It shows that when you're talking about different nutritional types of organisms, they get described based on their source of energy up here, where they get their energy, and there's a couple of different ways that they can get it, either from chemoorganotropically, which is when they get their energy from organic molecules, or chemolithotrophically, when they can get their energy from inorganic molecules. The third kind is one we're not going to talk about very much probably, and that is when they get their energy from light, which should be phototrophy, right? Any organism that gets their energy from light is a phototroph, and you guys learned all about that in probably Bio 121. We're going to be talking about when organisms get their energy from chemicals, either organic chemicals or inorganic chemicals. As far as their carbon that they need to build their metabolites and their macromolecules, they can either get it from CO2, and when that's the case, they're called autotrophs, or from organic materials like glucose, which is what you guys learned about over and over again. And in that case, they're called heterotrophs because they're getting it from a different place. And then the last category is electron sources. Those are mentioned up here already, where they get their electrons. So it's easy to get the energy and the electrons mixed up because sometimes we use electrons to make energy, to make ATP. But in this case, we're just talking about energy as reducing power. We're not really worrying about what it's used for. Organisms can either get them from organic molecules or from inorganic molecules. So there's a few examples. I don't think these pictures are in your book. But let's break this down a little bit. I would focus on this table, you guys. Table 11.1 on page 231. It basically just outlines the things I just told you, but this is a good place to refer to it so you don't have to write it down right now. The difference between autotrophs and heterotrophs, that's the easy one. Energy sources, either chemicals or light, also the easy one. I think if there's anything that confuses people, it's the source of electrons, because this is so different from what we usually think about. So there's some pictures here showing different categories of organisms that you might see. This one, pond, there's all kinds of organisms in here. What classes of organisms, using these words back here, would you expect to see in this pond? There's certainly phototropes, right? Photolithoautotrophs, that would be any kind of plant you've ever learned about before, would be a photolithoautotroph. They get their energy from the light. Where do plants get their electrons? This is what I would not ever think of as being a good electron source. Did you guys learn about this in Bio 121? Or anywhere? I bet you guys have learned it about 30 years more recently than I have. Here's a picture from the book. This book has like stuff in here that is like way beyond what I would ever get around to talking about microbiology. This shows photosynthesis in a thylakoid. And the source of electrons is down here, water, which is not something you usually think of as a source of electrons. They get their electrons from that. Some microbes can do that, get their electrons from water. It also could be, organisms could be getting their electrons from organic material, photoorganoheterotrophs. There are lots and lots of bacteria in soil in an environment like this that get their energy from the light, but they also can take electrons from organic material that's around them, also get their carbon from those organic materials, and that's really important for soil health. It's the kind of thing that can happen either in light or without light in decomposition processes. Here's an example of one of my favorite bacteria in the whole world. In fact, these are my bacteria. These are the ones that I got my PhD thesis studying. This is my old lab bench at the University of Iowa. The bacterium in those pictures is the color of cranberry juice. And they're called Rhodopseudomonas palustris. And they're purple non-sulfur bacteria. I just looked at the Winogrosky columns up there, and I can see some columns that have purple patches that are probably Rhodopseudomonas. These bacteria can grow photoorganoheterotrophically, meaning they're doing photosynthesis. They have different photosystems in plants. And they can get their carbon and their electrons from organic material, like succinate or acetate or other kinds of organic acids. They don't usually use sugar. They're not really into glucose and glycolysis. They do all kinds of other things that are important in the soil. Interesting thing about these, and to complicate your life a little bit, many organisms fall into different categories. So we said that these are photoorganoheterotrophs. These organisms can also grow photolithoautotrophically. So they can use up the carbon. It's really nice if they have fixed carbon and they're growing heterotrophically. Let me write that down so you can think about what all that might mean. Stupid thing. Photolithoautotrophy. If I put my hand on the screen while I'm trying to write, the whole thing stops. So what does this mean? Where would they be getting their electrons? These particular organisms can get their electrons from hydrogen, molecular hydrogen, which is a strong reductant. They get their, if it's autotrophic, they have to be getting their, I'm off the edge of the slide. They have to be getting their carbon from CO2 like a plant does. In terms of lithotrophs in general, lithotrophs can get their electrons. Let's go back a second here. Lithotrophs here, can get their electrons from inorganic electron donors. So some examples of inorganic, what would be an example besides hydrogen? We'll talk about some specifics about this later. Other examples of inorganic electron donors could include ammonium. We talked about this a little bit in terms of your Winogrosky columns. What did we say were examples of electron donors that participate in the cycling in your Winogrosky columns?
H2S.
Yes, H2S, which is hydrogen sulfide, or even elemental sulfur. Those are all examples of electron donors. So there's iron would also be one. You can imagine that pretty much any, and so the nerdiest of microbiologists, many that I know, make a hobby of trying to isolate organisms that can grow on different kinds of inorganic electron donors just to see if they can. So you would take media, give them no oxygen, usually they're grown anaerobically, give them no sugar or any other source of organic electrons, and just see if you can get organisms to grow on different kinds of inorganic electron donors. So here is another example of that. This is one that's showing, this picture in the book might have actually had the purpose of showing the internal membrane systems. Remember when we talked about the plasma membrane, we said sometimes there's extra membrane systems inside the bacteria? In this case, there's extra membrane systems that are used for oxidation of nitrite. So these are organisms that are getting their electrons from nitrite, NO2, and making nitrate, NO3. And they're part of a larger cycle. This is really, really important in terms of soil fertility and agriculture. If a farmer puts ammonia or anhydrous ammonia or nitrate on the soil, they want to keep it available for plants, but there's a bunch of organisms that are using it as part of their energy cycle that we'll talk about later. And this is an example of one of the organisms that participates in that. Here's another beautiful one you might see in your Winogrosky columns, Beggiatoa. It's a chemolithoautotrope. It gets its electrons from sulfur, its energy from chemicals, and it fixes CO2. So I know that so you guys are going to have to think about this some more. We'll talk about it again another day, but I would review these two tables and compare where the organisms are getting their energy and their carbon. We're going to just focus today on a couple of we're only going to get to one category today, and those are the chemoorganoheterotrophs. This thing is awesome. Chemoorganoheterotrophs. So you guys have learned about chemoorganoheterotrophy before. Let me go to the other slides here. There's a little duplication because I put these on for a different purpose at one point. We're going to talk about chemoorganotrophic fueling processes. And the two that you guys have learned about before are just in terms of the catabolism now, right? We'll talk about anabolism in a couple of days. In terms of catabolism, you guys before have learned about respiration and fermentation, right? You probably learned about it like 3 times before. How many of you took biochem? Yes, so not so many. You learned about it in cell bio. Did you learn about fermentation in Bio 121? So why don't we do a little searching of our brains and talk to your neighbor for a moment and see if you can come up with a definition for respiration. Cellular respiration. This doesn't have anything to do with your lungs. And fermentation. Tony, you can ask. I said, what were you going to ask? Oh. Oh, you got the definition? Hold on to it. I want everybody to see if they got it in their heads. Everybody take a second and talk to your neighbor if you need to. And then make sure in your definition it allows you to differentiate between the two.
Respiration.
Between respiration and fermentation.
Like oxygen, like something that doesn't have oxygen.
I'd say, yeah, like cellular respiration is where they would like take in something and then like you'd either like it's either like you take in CO2 or O2 and then like at the end you either get out CO2. Yeah, then they get energy. Yeah. And what was the last one? It said cellular respiration and
Fermentation.
fermentation.
Fermentation is when you don't have oxygen, but you need to produce ATP.
It takes sugar. And it takes the harnesses the energy from sugar for its energy source.
And then you make alcohol.
They make alcohol of the same product. That's how I know. I was thinking of the wine.
Or lactic acid in muscles. So
Yeah. Yeah, it makes sense. Yeah, generally you can touch the screen, it'll get bright, and then after a few minutes it'll get dim again.
Of respiration.
Aerobic.
It could be aerobic. What does it mean if it's aerobic? What is the oxygen? What's the role of the oxygen in aerobic respiration? It's a terminal electron acceptor. Yeah, what were you going to say, Ben?
I was going to say respirations when you have a final electron receptor.
It's when you have a terminal electron acceptor. That's true. And would that be true of fermentation? Tony?
The main difference is what happens to the pyruvate, which in fermentation, it uses the pyruvate to generate the ABH that it uses again for glycolysis.
There's a lot of good stuff in there.
And then
Let's decompress a second here. So in the context of fermentation and respiration, we're using sugar as your source of electrons, which not all bacteria do, but that's what you learned about before, is sugar. Glucose serves as the source of electrons, and you convert it into pyruvate, and then pyruvate can be the electron acceptor to do what now?
for respiration,
Yeah.
go into the that one side.
Yep, that's my favorite cycle. Krebs, I do have a favorite cycle. Sorry. Yeah, it goes in the Krebs cycle, and then, and what's the difference between that and fermentation?
Well, the products of that aren't used for glycolysis, but they're used to go into the electron transport chain.
For respiration?
Yeah.
Yeah. And then what you said before about fermentation is that pyruvate serves as a terminal electron acceptor to regenerate NAD plus for glycolysis to continue, right? So the difference between the two is, in both cases, you're getting electrons, and talking about glucose, you're getting electrons out of glucose, and in glycolysis, you're making ATP. With respiration, you have a terminal electron acceptor that is an exogenous. It's an external terminal electron acceptor that sits at the end of the electron transport chain. We'll talk about the electron transport chain later, but the big deal about that is it generates a proton gradient that can be used as energy, right? With fermentation, you have an endogenous electron acceptor, which in the case of glucose is pyruvate. So it's an organic electron acceptor that's part of the pathway that you're talking about. And the organic compound gets reduced instead of using the reducing power to drive the electron transport chain. And what can you say about that? Reducing a pyruvate, what do you get?
Or you end with lactic acid.
You end with, there's lots of things you can end with, but lactic acid is one of the examples. You end up with an energy-containing organic compound, right? So from the perspective of a cell, that would be wasteful because you're using something with value as a sink to dump something with value. See what I'm saying? We're going to talk about that more later. But you guys have got the basic idea that with respiration, you're using the electron transport chain, you're using some kind of exogenous thing. With fermentation, you're using an endogenous molecule. So both of those fit into chemoorganotropic fueling processes. Here's respiration up here. Organic energy and electron sources like sugar. Some bacteria use amino acids instead. We'll talk about that another day. The electrons are used for the electron transport chain. Here's the oxygen down here. Under aerobic conditions, they generate a proton motive force and make ATP by oxidative phosphorylation because, but we'll get to that later. Organic energy, go down here. It can also happen anaerobically. So this is one of the easy misconceptions that is there because you learned about it in the context of humans, is that fermentation, notice down here under fermentation, it doesn't say anything about whether it's aerobic or anaerobic. It just says that the energy comes from your organic compound. Here's energy generation by substrate level phosphorylation. That's the only ATP. SLP stands for substrate level phosphorylation. Do you want me to write that down? Substrate, I can't write on here. Substrate level phosphorylation. That happens up here too. but most of your ATP comes from oxidative phosphorylation with respiration. This doesn't say it has to happen under anaerobic conditions, nor does this say it has to happen under aerobic conditions. It just has to do with whether there's oxygen present as a terminal electron acceptor. So in some organisms, they ferment whether they have oxygen or not. That's just all they can do. So the reason that you have it, that we all learn that one is an alternative to the other has to do with human cells, right? And yeast. In humans, if you're exercising aerobically, you can do this, right? But what happens in your muscles if you run out of oxygen?
Fermentation.
Fermentation in your muscles, they break down glycogen, they make lactic acid, and they get all stiff and sore. Same thing happens with yeast, only they generate ethanol and CO2. In some organisms, they have just this or just this. These are all different options that don't necessarily occur based on the presence of oxygen or not. So we're going to talk about a little bit about how that works and the diversity of the different ways that that can happen. And we're going to talk about energy generation, not just from sugar, which you've learned about before, but all kinds of molecules. So microbes in the environment are responsible for the recycling of all kinds of material. So anything that ever lives dies and gets broken down by microbes. So that includes not just sugars, but all kinds of other things, including proteins, which are shown over here. Proteins get eventually broken down into amino acids that get funneled into the central metabolic pathway. So the top of this shows glycolysis up here. There's a conversion of pyruvate to acetyl-CoA here, and the citric acid cycle, or this book calls it the tricarboxylic acid cycle, or the TCA cycle. It's also known as the Krebs cycle. So I know you guys have learned about this several times before, but it's something that bears repeating. But I would, if I were you, I would review, this is the figure for glycolysis. I would review this and I would focus on, we're not going to talk about every single enzymatic interaction and the investment stage and everything else, because I know you've learned that before, but review that and be sure that you know what you get out of glycolysis. So the equation for glycolysis, and that's on the bottom of page 236, in case you don't still have it memorized. Page 236. Because we're going to be comparing that to some other things. Okay, so sugars, proteins, phospholipids in the environment are also broken down by bacteria. Everything that ever lived, which would include lignin, right, from plants, lignin also is broken down, and it also bypasses glycolysis and would come in down here, essentially. So we're going to try to talk about some of the ways that energy gets extracted by things besides glycolysis. At the same time, bearing in mind that it's easier to learn if you can compare and contrast things. So since you're familiar with glycolysis already, it'll be helpful to think about that as an example so that you can compare new information to that. So glycolysis, I'm going to put that down in your notes. You're going to review page 236. How many of you guys still have it memorized from some other class, what you get out of glycolysis? Tony does.
Two ATP and two NADH.
Well, what's the organic?
NADH.
NADH, and the most valuable part of it is pyruvate, and two pyruvates. There you go. So easy. So that's glycolysis. I want to talk about another pathway that not every organism in the world has glycolysis, which is bizarre. And the reason that they can get away with that is because there are other ways to get energy out of sugar. It's important to remember, though, that there are times when an organism needs some of the intermediates in the pathway. And so the glycolytic pathway, which is abbreviated here, is what's called an amphibolic pathway. And we talked about amp is something earlier, right? What does it mean that it's an amphibolic pathway?
It has polar and nonpolar properties.
That was a phospholipid. That's the amphipathic molecules. You're on the right track. Meaning it has both properties. So how does that apply to this?
It has catabolic and anabolic.
It has catabolic and anabolic properties. So some organisms don't have glycolysis at all, but because they have some of these enzymes that are amphibolic and can be used for anabolism, they can still make the intermediates they need. And so an example of an alternative pathway is this one, the Entner-Deuteroff pathway. Make sure I spell Dr. Deuteroff's name correctly here. So this is another glycolytic pathway. You could say that this is glycolysis. So how are we going to tell the difference between what you call glycolysis in cell biology and this glycolytic pathway? Did you guys learn another name for glycolysis? It's also known as the Embden-Meyerhoff pathway. Does that sound familiar? No? So glycolysis, there's two F's in Meyerhoff. The glycolysis that you guys learned about before with the 2-pyruvate and the 2-ATP and the 2-NADH, that is also known as the Embden-Meyerhoff pathway after the guys who worked it out. This other glycolytic pathway is known as the Entner-Deuteroff pathway. And it's found, it's not as common, but for some organisms it's especially important. For example, soil bacteria. Typically, the Entner-Deuteroff pathway is found in bacteria that are soil bacteria. And what do you get from it? From the Entner-Deuteroff pathway, which is shown up there. It utilizes some of the same enzymes that are in glycolysis down here. But overall, you take glucose, I'm just going to cut to the chase here. You get 2-pyruvate. You get 1 ATP. I'm going to put 1 here because that's a difference. You get 1 NADH and 1 NADPH. So the energy-containing products, or the usable energy. So is pyruvate an energy-containing product? Would you call that an energy-containing product? Yeah, that's the most valuable. In terms of any organism that can respire, this is where most of the energy still is, so that's definitely an energy-containing product. In terms of the other products, you get half as much ATP, and you have your NADH and NADPH split up between the two. So what is the, who cares about this anyway, right? Because we just step on the bacteria that are doing it. What can you predict about the bacteria that use this pathway instead of the Embden-Meyerhoff pathway? Would you think it's a very important pathway to them, Ben?
Well, I would say it's maybe in relationship with plants, because plants use NADPH instead of ATH. I don't know.
Well, they are in the soil. I think that the thing about being in relationship with plants is true in the sense that the bacteria that are doing this probably don't put such a great value on glucose, because otherwise there would be a selective advantage for them to have the Embden-Meyerhoff pathway, right? Because they get more energy out of it. They get more twice as much ATP out of the glycolytic Embden-Meyerhoff pathway. And so it's true that as plants break down, they release lignin. So a lot of the bacteria that live in soil that would have this are probably not ones that are specializing on growing on sugar. So what would you think about the bacteria that live in your guts and your mouth? They're probably going to have the Embden-Meyerhoff pathway because they're using sugars that they get from us. Whereas the bacteria in soil that are decomposers, they might not rely on it as much. The other thing, what's the big deal about NADPH versus NADH?
It's not used in anabolism or anabolic pathways?
Yep, NADPH is used in anabolism. So another advantage you get from this pathway is that you get a source of energy for anabolism. They don't get as much ATP, but they do get NADPH. So would this be advantageous? It just depends on where you live, right? So in the end, the organisms that have this anabolism, they're not as interested in ATP, so they don't care if they only get one. If they are if they're respiring organisms, the difference between 1 ATP in Embden-Meyerhoff and Entner-Duderoff isn't that big of a deal anyway, right? Why would it be insignificant for the respiring organisms? Based on what you learned before about Krebs cycle, Luke?
You're still gonna get 30 ATP roughly from.
You're going to get a whole bunch of ATP from the electron transport chain, so it's not that big of a deal. So why would one organism have one over the other? It's not like they get to pick, it's based on what genes they have. They have the pathway for this, or they have the pathway for that. There are some of them that probably have some enzymes. In fact, they know that there are organisms that have some overlap and have some of the enzymes for both, and they're regulated in different ways. But this is an example of the kind of diversity that exists just in terms of using one particular carbon source. Okay, so what happens, let's talk about the value of what we're getting out of this. So the value in terms of both of those two pathways for an organism that can respire is still tied up in pyruvate, right? So you got either 1 ATP or 2 from one of those two glycolytic pathways. There's still a whole bunch of energy that can be used in pyruvate. And this is why the organisms that respire have such an advantage over those organisms that ferment. So the respiring organisms can take the pyruvate and extract more electrons out of them by putting them through a pathway like this, through the TCA cycle, or also known as the Krebs cycle. So I'm a little bit hesitant to even show all of the structures on here because we're not going to memorize all of the reactions in here. That would be like a biochem class or something. But we're talking about it in the context of how does an organism, a bacterium, get its energy today. We're going to talk about it later in terms of the amphibolic nature, or the amphibolic nature of the pathway. So here's the pyruvate. It can be converted into acetyl-CoA right here. So things to know. This is the substrate for going into the entry point in terms of catabolism. The cells convert pyruvate, so this is the Krebs cycle, aka the TCA cycle. Acetyl-CoA enters. That comes from pyruvate, and the cells get a little bit of reducing power right here from doing that. And then the whole purpose, what would you say in the terms of catabolism? What is the overall purpose of this Krebs cycle? Well, what is the overall purpose of anything with respect to catabolism? Getting energy, yep. And so the energy that you get from the Krebs cycle is mostly in the form of reducing power. NADH here, NADH here, and FADH here, FADH2. So acetyl-CoA enters, you get the reduction power out, which means that the substrate is oxidized, right? Oxidation of the carbon. That gives you NADH and FADH2. 2 NADHs actually.
You get
What? Did I miss one of them? Yep, I missed this one up here. 3. See anything else I missed in there? Right. So you are so this is where further oxidation is occurring of the carbon. Other cool thing about the cycle. So this cycle is never mind. I love the Krebs cycle, but the cool thing about it, one of the cool things is the whole thing regenerates. So not only do you get to wring all the electrons out of there, it regenerates the accepting molecule up here. Oxaloacetate gets regenerated, so this thing can continue and take up 2 more carbons from acetyl-CoA. So oxaloacetate gets regenerated. So if you think of this strictly in terms of catabolism, you would have to go through the cycle 2 times to use up the glucose, to use up the carbon that came from glucose, right? So you start out with a 6-carbon molecule, you use one of the carbons right here after it's split it into pyruvate. What's the deal with that big line? take the 2, 3 carbon molecules that you got. Oh, I know what it's doing. Never mind. You take off one of them as CO2 up here, right there. So you have 2 carbons enter. So this cycle has to go through 2 times in order to fully oxidize the carbon, right? If you were assuming that you're using up all of the carbon from glucose for making energy, you'd have to run the cycle twice. So that's not necessarily a useful assumption that you would use it twice, because when microbes are growing on glucose like this, they're typically using the intermediates from the pathway for other things, which we'll talk about later when we talk about anabolism. So the electrons come out of there, and where did the electrons go? Should we talk about the total yield here? Let's just say electrons in the Krebs cycle are available for the electron transport chain. For respiration. So we're not going to talk about this right now.
Dr. Eglin?
Yep.
So the CCA cycle in bacteria, the lack of mitochondria, is this happening like right inside the plasma membrane?
It is, some of it's happening in the cytoplasm and some of them are associated with the plasma membrane, because some of these, look at this, here we go, it's this one. This reaction from succinate to fumarate is catalyzed by an enzyme that's associated with the plasma membrane. So we don't have mitochondria in bacteria, right? So some of it's in the cytoplasm and some of it's with the plasma membrane. That's right. So how many of you guys, so how would you have done if you just took a quiz on glycolysis and the TCA cycle? Because every year I feel like people don't look particularly like this is earth-shattering new information, but at the same time, I know when I was a student, I had to hear this like 2 or 3 times before I even got it. Was this a useful review? Next year, should I just skip it, put it on the test, and just say, remember when you learned it yourself? No? Do you guys have questions about, like, so this figure is in here, we're going to skip it. I'm going to spend time teaching you something different. Questions about glycolysis? Let's start out with questions about the concepts, and then questions about, do we have to know blank? Questions about the concepts. This shows funneling reactions of all kinds of molecules into the TCA cycle. This is just for the purpose of catabolism. We're going to talk about proteins and phospholipids and lignin and stuff another time. Questions about this? Amphibolic nature, this is going to be more related to when we talk about anabolism later. But for example, glucose 6-phosphate, some of this stuff is really important for making your nucleotides. And if you don't have the Embden-Meyerhoff pathway and you don't use glucose, how are you going to get it if you don't have an amphibolic pathway that can take, imagine if you're a bacterium and you're eating pyruvate, you need to make this stuff, and so these reactions are used this direction. A lot of gory detail. We talked about substrate level. So how is the ATP made from glycolysis? It's all substrate level phosphorylation, right? There's the investment stage, there's the return stage, which is done by substrate level phosphorylation. Entner-Duderoff pathway. What's the coolest aspect of the Entner-Duderoff pathway? Pretend you think it's cool for just a moment.
Makes NADPH.
You get NADPH, which is different from glycolysis. That's probably the highlight. Also that some organisms in soil have this instead of glycolysis, even though all you ever learned about was glycolysis. Oxidation of the carbon that comes in, formation of 3 NADH and 1 FADH2. There's also substrate level phosphorylation going on down here that we don't talk very much about. There's 1 ATP equivalent down here. Why don't we talk very much about that? In terms of importance and energy balance. Ben?
Because you get way more from transport chain.
Yep. That 1 ATP is pretty insignificant. You have a question?
Yeah. Could you go over to the endogenous electron acceptors one more time?
Yeah. So, we're going to talk more about fermentation another day, but imagine you're using this pathway to get energy. And if a cell is going to ferment, the electron acceptor that's going to get used is an organic molecule within the pathway, pyruvate, because it's endogenous, meaning it's inside the pathway. If the organism is going to respire, it's going to get something from outside of itself, like an inorganic compound like nitrate or oxygen or something that's coming from outside of the pathway and outside of the cell. Other question? What's the deal with this? Why would they even stick this in here? It has to do with what is a good electron acceptor and how the electrons flow through here, which we haven't really got to. We're not going to go into all the detail about that because it's way more interesting to talk about bacterial physiology in ways that are unique. So this is actually, everybody good with that other stuff? Okay, this is about the actual respiration part, and the book says this figure is actually based on mitochondria, and they're showing it as kind of a reminder and a comparison of how similar the bacterial respiration is to what happens in mitochondria. So here's NADH down here. This came maybe from the Krebs cycle. A bunch of it came from the Krebs cycle, and the electrons from this are used to reduce the electron carriers in here, which are represented as these big blue blobs. And some of them are proton pumps. And as they get reduced, they pump protons outside the membrane. So this is the plasma membrane. Whoops, this is not the plasma membrane because we're talking about a mitochondria. This is the intramembrane space. And the protons then are used to drive this ATP synthesis using ATP synthase, right? That's happening in mitochondria. We talked about another use for the proton motive force the other day. What was that?
Flagellar movement.
Yep, Ben.
So in bacteria, instead of pumping the protons into the membranous space, they'd just be pumping it out of the cell entirely?
Yep, they'd be pumping it into the periplasmic space. So here's what it looks like in a bacterium. They're pumping them out into the periplasm. And in this example, they're showing something a little bit different here. One of the points that they're making with this figure is that there are alternative places that an organism can get electrons. So this is a bacterial cell membrane. The book says it's Paracoccus denitrificans, which is an organism that we'll talk more about later that can also grow anaerobically. But it's showing aerobic respiration with electrons coming from NADH right here, protons getting pumped out here, protons getting pumped out here. And what about this? This is showing a little value added that this particular organism can use. What is this molecule? Methanol, right? It's methanol. Some organisms can get their electrons for respiration from weird places, things we would consider weird. So this is an example of an organism that's using methanol as part of its respiratory system. So it's pumping protons out here. Some of the electrons that are used to drive this come from other places. And what's not shown is it's got the same ATPase, very similar, making ATP, as what you see in mitochondria. Only in this case, it's localized to the plasma membrane. So this is still aerobic respiration, even though it's doing something weird with methanol and using that as a source of electrons. So this figure, we're not going to, let's skip this figure. The point of it is just to show that in bacteria, there's different kinds of systems based on how much oxygen is available, because there's different versions of the enzymes that have different energy potential. So imagine if you have an organism that is growing chemoheterotropically. It uses up all of its oxygen. Many of the bacteria that live in the soil have the ability to use alternative electron acceptors besides oxygen under anaerobic conditions. They're still respiring, but they are just able to switch to another electron acceptor. So what would it take? Imagine, look back at this figure. What would it take to use oxygen, or use something other than oxygen right here? It seems like an amazing property, but all it really takes is a different enzyme that reduces some other terminal electron acceptor. So here's an example. So you guys should know some examples of these. Terminal electron acceptors used for anaerobic growth. Nitrate, sulfate, those are super common. And some of these are actually pretty good in terms of energy. So a lot of bacteria can use nitrate as a terminal electron acceptor and grow really, really well. So when you learned about yeast and growing anaerobically, You probably learned that they grow really well if they have oxygen and they can do respiration, and then they switch to fermentation and they grow kind of crappy because they don't have as much energy. If an organism has the ability to switch from aerobic respiration to anaerobic respiration, it doesn't necessarily grow crappy, depending on what its terminal electron acceptor is, because this is still pretty energetically favorable to use nitrate. So they can use nitrate. There's a figure coming up here that shows an example of that. So this, let me tell you what organism this is, because we're going to go hear it again another time. Oh, this is Paracoccus denitrificans again. It's got the name denitrificans because it denitrifies. So here it's getting its electrons from NADH. Here it's using nitrate and is reducing nitrate with one of the electrons to make nitrite right here. And this nitrite can come up here and get used again, gets reduced and reduced again to make nitrous. This is nitric, right? Nitric oxide. This is nitrous oxide, which is then reduced to make nitrogen gas. So it takes different enzymes to reduce all these different nitrogen-containing compounds, but in the end, it still achieves the same kind of an outcome. It starts with nitrate and reduces that. Instead of using oxygen and making water, it uses nitrate and makes nitrogen gas. So some bacteria can't go all the way from nitrate to nitrogen gas, and they go different steps along the way, and we'll talk about that when we talk about the nitrogen cycle. One quick question is, we have one useful minute. Does anybody have any questions about, so what's the difference here? There's different carriers, different enzymes. So you know these globs represent enzymes, right? They're enzymes that are taking the electron from one place and putting it into another place. You'll learn all about the details of that in biochemistry. Anna?
You said there's independent reading for chapter 7, but we can't see that.
I'll put that back up for you. Yep, thanks. There's also, there'll be some others too. Okay, somebody's presenting on Friday, right? Nobody presented today, but there'll be somebody Friday. You guys would serve yourselves well to review respiration, review glycolysis.
Okay.
Know the products of glycolysis.
It's really weird.